Hey everybody, Dr. O here, and in this video, I want to talk about the oxygen dissociation curve, and specifically, what's happening at the tissue level that makes changes in this curve. So, let's get started. Now, if you're studying the lungs or respiration or blood saturation levels, you're going to see a curve like this, and this is called the oxygen dissociation curve. And this curve is telling us about the relationship between the amount of oxygen in the area and how much of that oxygen is showing up in the blood. So if we look along the bottom line, the x-axis, we can see that that is telling us about the partial pressure of oxygen, which is how much oxygen there is. We can almost think of that as the concentration of oxygen molecules in a particular environment. Now on the y-axis, we're going to have the percent saturation, and what that's telling us is how much oxygen is in the blood. So for example, if we had a partial pressure of 40, then that would tell us that we can expect to have about 75% saturation of oxygen in the blood. And this is what we see with deoxygenated blood that is returning to the lungs to get filled up with oxygen and drop off carbon dioxide. If we look at the other end of things, if we had a higher uh, uh, partial pressure of oxygen, like maybe 90, then we would expect to also see a higher percentage of blood in oxygen in the blood. And and indeed we do. We can see that when our partial pressure of oxygen is 90, then our percent saturation of the blood comes in at about 95. So this is telling us about the relationship between pressure of the oxygen in the environment and how much of that we can expect to find in the blood in a value that we call percent saturation. Now, often there's a little bit more detail that we see in these charts, and it looks like this. So then the question becomes, what does this curve and that curve tell us? Well, that's what we're going to talk about in the rest of this video. In this video, we're going to be looking at things at the microscopic level, at the tissue level, trying to understand the factors that are going to influence oxygen's behavior, whether it's going to be tightly attached to hemoglobin or whether it is going to unattach to hemoglobin. And in order to do that, let's bring up a hemoglobin molecule and talk about its structure. Now, hemoglobin is a protein. It's a quaternary structure. It's made up of 4 strands of protein that have come together to create this functional molecule. Now, in the center of every single one of these strands is a portion of the molecule that's called the heme group. And in the center of the heme group, we're going to see that we have this little nugget of iron. And that nugget of iron is going to be the attachment site for oxygen. Now, instead of demonstrating the hemoglobin as this molecule that is moving around the body, instead I'm going to demonstrate it as the hemoglobin bus. And this bus has 4 seats, which are the 4 irons that this molecule can offer up to oxygens, and those are going to be the targets for the oxygen. So here we are. The hemoglobin bus has arrived at the lungs. We've just taken a deep breath in. Our lungs are fully oxygenated, and oxygen is going to be compelled to come and take a seat on the hemoglobin bus. Now, once the blood has been oxygenated, once the hemoglobin inside of the red blood cells has been oxygenated, then that hemoglobin is going to be ushered out to the tissues where maybe the oxygen will come off. So let's do that. When the hemoglobin bus pulls up to the tissue, the question becomes, what will happen to oxygen's affinity for hemoglobin? Or another way we can ask that question is, will the oxygen get off the hemoglobin bus or is it going to stay on? And that's going to really depend upon what's happening out in the tissues. So when the hemoglobin bus pulls up, the oxygen molecules on the hemoglobin bus are going to look outside and see what's going on. And if one of the things that they see is a lot of other oxygen molecules out there in the tissues, then they're going to determine that it's too crowded for them to get off the bus. So keep in mind that when gas molecules are looking at concentration gradients, they're only looking at similar molecules. So oxygen molecules are only looking at oxygen molecules. They don't care what other type of molecules are out there. So that's why we call it the partial pressure. And that's because molecules of gas are only concerned with other molecules like themselves. Okay, so they're looking at the population of the other gas molecules in the area to determine if they want to get off or if they want to stay on. And so in this case, too much oxygen out in the tissues is going to be a trigger for the oxygen molecules to stay on the hemoglobin bus. So what else are they looking at? Well, another thing that they're going to be looking at is pH. Now, oxygen out in the tissues is associated with alkalinity. So when the oxygen molecules on the hemoglobin bus look at the pH meter and see that it's alkaline, that is also another indication that the oxygen on the hemoglobin bus needs to stay where it is. Because basically that's also telling it that there's a lot of oxygen in the tissues. The third thing that the oxygen molecules on the hemoglobin bus are going to look at is temperature. And if the temperature is a little too low, then that is also another indication that the oxygen needs to remain on the hemoglobin bus. Now, why is that? Well, usually if cells are not that metabolically active, they're not making a lot of chemical processes occur, which typically generate heat. So if they're not using oxygen to make maybe ATP, for example, they're not generating a lot of heat, and that can lead to a lower temperature in the environment, which is basically further confirmation that the oxygen on the hemoglobin bus needs to stay where it is. So with all of these factors in place, that is a lot of oxygen in the tissues, a high pH, and a relatively lower temperature, the oxygen on the hemoglobin bus is basically going to bypass this tissue and go find some other tissues where maybe the oxygen will then want to get off the bus. So let's do that. Before we move on to another scenario, let's just analyze what took place at the tissues and relate it to this chart. So one of the things that we recognized at the tissues is that there was a low level of carbon dioxide molecules, which meant that we had a lot of oxygen molecules, because typically the two are related. When one level is high, the other one's going to be low. The other thing that we saw was happening at the tissue level was that we had a high pH. We were alkaline. The third thing that we took a look at was the temperature, and we saw that there was a relatively lower temperature than anticipated, and that all three of these basically caused the affinity between hemoglobin and oxygen to be higher, which meant that oxygen is going to stay on the hemoglobin bus. And when we see this scenario playing out, that basically means that this graph that we're looking at is going to shift to the left. So it basically means this line is now going to shift this way. And this is the part of this graph is telling us that there is a relationship between the pressure of oxygen in the area and the amount of oxygen that's going to be in the tissues or in the blood, and that can cause this graph to shift. So it's a dynamic relationship between oxygen in the environment and the ability or the inclination for oxygen to either remain bound to hemoglobin or to release from hemoglobin. And in this case, because we had the right set of factors in the environment, oxygen is going to stay strongly bound to hemoglobin and remain on the hemoglobin bus. So we're going to look at a different scenario this time, and we're still going to ask the question, what will happen to oxygen's affinity for hemoglobin? Or put another way, will oxygen get off the hemoglobin bus, or is it going to stay on? Well, in this scenario, we're going to see that the tissues are populated with a lot of carbon dioxide, but not very much oxygen. And remember, oxygen molecules are only concerned with how many other oxygen molecules there are in the area. And when there's low oxygen molecules in the tissues, that's going to be a sign that the oxygen molecules on the hemoglobin bus can depart. And so they do. Another thing that the oxygen molecules on the hemoglobin bus are going to be looking at is pH. And we know that we can associate carbon dioxide with acidity because it has a tendency to make carbonic acid. So we take carbon dioxide, put it in the environment, and it will combine with water and turn into carbonic acid. And that's going to make the environment acidity. And that is another indicator to our oxygen molecules on the hemoglobin bus that it's okay for them to get off. And so they do. The third thing that the oxygen molecules on the hemoglobin bus are going to be looking at is temperature. And here the temperature is going to be slightly elevated, and that is going to be another indicator for the oxygen molecules to get off the hemoglobin bus, and so they do. So again, taking a look at these factors, it helps us to understand the why behind them. So what's going to move oxygen off the hemoglobin bus? Well, we're going to have low oxygen in the tissues, high oxygen on the hemoglobin in the bloodstream, and that's going to create a favorable concentration gradient for oxygen to leave the hemoglobin bus and get into the tissues. Acidity is an indicator of the presence of carbon dioxide. And that too is another indicator of low oxygen in the tissues, which is an indicator of the appropriate concentration gradient to drive oxygen off the hemoglobin bus and into the tissues. And then temperature basically tells us that we've got high metabolic activity. The cells at the tissue level have used up the oxygen that was available to them, produced this byproduct carbon dioxide, which is now populating the tissues, and in the process generating a higher temperature. And this is another signal to the oxygen on the hemoglobin bus that it's okay to depart. So if we take the information that we just looked at in the previous scenario, where we had high carbon dioxide levels and low oxygen levels, low pH or acidity, and a higher temperature, these are the conditions wherein we're going to find that hemoglobin and oxygen have lower affinity. They're less likely to remain bound together, which means that oxygen is going to leave the hemoglobin bus. And on this chart, that's going to cause a shift to the right, which means that this line is going to shift to the right. To recap what we learned about the hemoglobin-oxygen affinity, that is, whether or not oxygen is going to leave the hemoglobin bus, we learned that the curve that we looked at will shift to the left when we have low carbon dioxide or high oxygen levels in the tissues, when we have a high pH or when the tissues are slightly alkaline, or when we have lower temperature. And the way that we would say that is that we're going to have a higher hemoglobin oxygen affinity when these things are in place, which in our video was translated to oxygen staying on the hemoglobin bus. Now the curve is going to shift to the right when we have high carbon dioxide levels or low oxygen levels in the tissues, when we have a low pH or when our tissues are slightly acidic, and when we have higher temperatures. And we would say that we have a lower affinity between hemoglobin and oxygen, which in our video meant that this is the right time for oxygen to get off the hemoglobin bus. And with that, we are done. Thanks for watching.